sponsored by the MRC Centre for Medical Mycology at the University of Exeter. My name is Sarah Gaffen. I'm from the University of Pittsburgh, and I'll be your chair today. My co-chair is Jay Coles from Tulane University. And we're really thrilled to have as our inaugural speakers today, Arturo Casadevall and Rebecca Drummond. Dr. Casadevall is a professor and chair of molecular microbiology and immunology at Johns Hopkins University. Rebecca Drummond is assistant professor and MRC Career Development Fellow at the University of Birmingham. This series was the brainchild of several of us researchers in the medical mycology community, and our international organizing committee that resulted from those early conversations is listed on this slide in the blue box. Within just a couple of days of announcing this on Twitter and our professional networks, we were really excited but kind of astounded that the 500-person limit for viewing these was completely filled, thus supporting our feeling that there was a real hunger for this kind of series in our research community. So we're delighted that you're all here, but never fear, anybody who missed it can see these talks on the YouTube channel for an extended period of time. The link to that is on the MRC website, which is listed at the bottom of the slide. And we encourage you to send anyone who missed it to that site so that they can see these talks. Every talk is going to have two speakers, two 30-minute talks, and we will reserve all questions to the end. Questions can be taken by using the Q&A feature on Zoom. Please do not use the chat. That will not be moderated. We will not see questions on the chat. So use the Q&A. Those questions will only be visible to the moderators, and we will have plenty of time at the end to take questions. If we don't get to your question for time, we apologize, but hopefully it's the beginning of just a lot of exciting conversations. Our goal in these talks is to achieve diversity, both in career stage, geography, gender, field, etc. Okay, so the upcoming MycoTalks are listed on the website. Again, the link is at the bottom. And our talks for the 27th of August and the 24th of September are listed here. Please remember you need to register for every session individually. So please do that. You can get email alerts by connecting again on the website and signing up. You can follow us on Twitter. The hashtag is indicated there. And so given the popularity, please sign up quickly when you get those notifications. Finally, I really want to thank the crew at the University of Exeter who's been working behind the scenes to make this run smoothly, including Jules and Lauren and Alberto, and they've really been wonderful to work with. So with that, I will end my talk and take us to our first speaker, Dr. Casadevall.
So hopefully this is showing, but a little worried that this is. So Sarah, I assume you can see everything?
Yes, we're good.
Okay, good. So I'm gonna have a fun, it's a fun talk. It's updating the fungal infection mammalian selection hypothesis at the end of the Cretaceous period. So around circa the turn of the century, there were three questions that bothered me. Why are there so few fungal diseases for humans? We have dermatophytes are common, but invasive disease is rare. The big ones are Candida, Cryptococcus, Aspergillus. But, you know, these are very few if you compare it to the number of bacterial or viral diseases. Why are mammals so resistant to fungal diseases? Fungal mammalian is rare unless you have impaired immunity, but immunity can't be the sole answer because all vertebrates have adaptive immunity, but reptiles and amphibians are susceptible. And that leads to the sociology question: why is medical mycology so unappreciated relative to bacteriology, parasitology, and virology? And the answer is it's late, summer, and we just don't, the fungal problem in humans is not as big as in the other disciplines, although that certainly changed at the end of the 20th century. So my approach is read outside your field. Think in terms of evolution, think of mammalian selection. And when I began reading, I was bothered by none of the mammalian emergent hypotheses considering infection. And in fact, none of them made a lot of sense to me. There was no good explanation for how mammals became dominant large animals. Began to think in terms of deep time. Deep time means not yesterday, not a month ago, but in terms of millions and millions of years ago. Embrace holism. We're all trained to think in reductionism. You know, you want to get to mechanism, you want to get to causes. This is going to require putting facts from very, very different types of areas together. Avoid the whole of reductionism. You can easily get lost in one thing that doesn't fit, but aim for an explanatory synthesis and fill in the details later. So this is an example of something that I would have run into at the time, reading outside my field. The platypus gets disease. The platypus is a primitive mammal. And here is a hint that it is, temperature is important. The temperature of a platypus is about 31 degrees, 32 centigrade, and they are quite susceptible to some fungal diseases. And even here in a box, susceptibility maybe due to the platypus having a body temperature of 32, while the maximum temperature of growth for this mucor is 36 degrees. Hints. So the age of mammals begins after the KT event. The KT event is the change between the Cretaceous and the world after that. And we'll talk more about it, but from a fossil record, the big difference is that you see a great change in the megafauna. Before you had a reptilian megafauna, and afterwards you had a mammalian megafauna. When you travel around the world and you see large animals, they tend to be birds or mammals. You don't see large reptiles. So what you see is that you have had a change between organisms that were ectotherms to organisms that were primarily endotherms. The birds are hot, the mammals are hot. So when you look at what happened at that time, there are a lot of theories. Some of them are funny, some of them were embraced. Clearly, smoking is probably not related, and I don't know what to say about the biblical explanation. But for many years, it was thought that it was small mammals that ate the eggs, and believe me, that makes no sense either. There is this paper that was published in a cell-press journal, Current Biology, arguing that they could have passed enough methane to cause climate change. Well, I leave that up to you, but to think about. But the two major hypotheses that are out there that are viable is that there was a lot of volcanism at the end of the Cretaceous. The Deccan traps, which are currently in India, that's what's called Decca, erupted, and that would have caused major climate change. And then there was an asteroid hit in the Yucatan, and the asteroid hit is increasingly being seen as the final hit that led to the extinction of the dinosaurs. But that raises a question. We have reptiles today. Why didn't we have a second age of reptiles? Why didn't the survivors just pick up and give us age of reptiles version 2? And I would argue that, you know, a reptile needs 1/10 the energy of a mammal. A reptile replicates much faster. They don't bother taking care of their offspring. So they should have had a huge survival advantage relative to mammals, and yet that didn't happen. The cost of the mammalian lifestyle is extremely expensive. If this is a log plot, and look where you are, you're at the top of the right part of this. The amount of energy that is required for you to get through the day is enormous. And consequently, mammals were a very minor group of organisms until 65 million years ago. And then you had the great mammalian radiations that gave us humans, whales, monkeys, horses. And I am very interested in how this unfavorable lifestyle becomes so dominant. In fact, there has to be something that's selected for them. Because to tell me that you're going to end up with organisms that need 10 times more energy than the others certainly doesn't really make a lot of sense to me. And so I thought the explanation had to be in the distant past. So let's talk about the Cretaceous world on its end. it was a warm, volcanic, and forested planet. There were forests in Antarctica. The Arctic was tiny, and I'm sorry, the Atlantic was tiny, and the rock hit in the Yucatan. It was populated by an enormous reptilia megafauna, and this is the area in the Yucatan where it hit. And if you want to know where the rim is, all you have to do is go visit one of the cenotes. If you go, for example, to Cancun, you can, the excursions to take you there, and people go there because they are the Mayan sacrifices places. But in reality, they're the only place in the world there are cenotes. And that's because the limestone melted and cooled very rapidly and created this set of channels. The post-impact world was a terrible place. You basically had fire, smoke, and dust obscure the sun. Photosynthesis is shut down at a minimum of 6 months, a maximum of 10 years. Think about who's going to survive in that calamity. And global temperatures drop by 10 to 12 degrees Celsius. This is global, in a period of a week. This was a world in which there is fossil evidence for a major global compost. You can find the layer, the fossils of fungi who reproduced to enormous numbers at that time. So you had all the conditions for a global fungal compost: cool, dark, lots of decaying vegetation, and there would have been massive inocula in spores. And one of the things we know is that even hosts can be overwhelmed. An intact host can be overwhelmed if you give them a massive inocula. But in this case, the survivors would have all been in trouble. And I will say more about the special troubles that plague the reptiles relative to the mammals. But endothermy, being hot, would have provided a tremendous defense against the fungi. This is before this, I drew with crayons, and this is a watercolor of the time. But the point that I want to get is, it's a dark, cold world that is being devastated. The post-impact world, so the reptilia megafauna, anything at the top of the food chain would have been hit. You had the impact blast, the disruption of food sources, the cooling of the planet, maybe fungal diseases. The other thing that is apparent now is that there were massive tsunamis set up by this. Mile-high tsunamis swept through North America and would have drowned enormous numbers of species. Reptiles fight infections, but there was no sun. There was no sun in many places, as long as a decade. Reptilian eggs are susceptible to fungus diseases from the outside, and we know that dinosaur eggs were susceptible to fungi because you can find today dinosaur eggs that are infected with filamentous fungi. So I was thinking about this, and I gave a talk in Miami University in Ohio, and Nicholas Money, who was the editor of Fungal Genetics and Biology, told me to write it down. So I sat down and I wrote it down. I had a lot of fun. And this was a paper that was published in 2005. And pretty much try to link these ideas. So the hypothesis is simple. People, by the way, get it wrong. They say Arturo says that the fungi killed the dinosaurs. No, a lot of things kill the dinosaurs. What happened is the fungi provides a fungal filter that selects the mammals over the reptiles, and that therefore we end up with the age of the mammals. And here is the KT boundary, and then you have the two ages separated. So what happens in the next 15 years? Well, the paper sits out there. Many of you have heard me give this talk. I go around and talk about it, and I keep thinking. I like to find examples and experiments that support the temperature and fungal susceptibility notion. You try to get experimental data where feasible. You keep up what's happening in geology and planetary science. And the hypothesis opens up new ideas and possibilities, especially in an era of global warming. So the white nose syndrome appeared in 2006, and it fit very nicely into this because you had a situation in which the bats were resistant in the summer and susceptible in the winter, when their temperature dropped to about 10 to 12 degrees and they become susceptible to a fungal disease. You can take the bats into the lab, feed them, make them warm, and they can clear the fungal infection. So this begins to fit within this pattern of resistance as a result of temperature. We began to get data. So I worked with Vincent Robert, and we looked at the collection, at the CBS collection for susceptibility to temperature. And what you can see is that most fungal species do well to about 30. But once you enter the 30s, their ability to replicate drops very rapidly. So in fact, for every degree in this range, you can exclude 5% of fungal species. Then I work with my friend Aviv Bergman, and he takes this data and then the cost of being hot, which is a well-known formula from mammalian physiology, and he solves what is the optimal temperature. And the optimal temperature that falls out is 37 degrees. Again, a piece of data, but it fits very, very nicely with this, suggesting that your temperature was selected for potentially as a trade-off between the number of calories that you needed to eat each day and protection against fungal diseases. So the first update of this is in 2012. Joe Heidman hears me talk about this and he says, you know, time to update it. So we added all the stuff that came in after 2005, and this was published at the time. Notice that I'm now focusing on the rise of mammals to try to avoid the misconception that Casadevall says the fungi killed the dinosaurs. They may have contributed, but I think there were plenty of other causes to knock them off. So then comes the question. Could people say to you, how do you know that there are fungal pathogens in the Cretaceous? Well, you know that the fungi are old. It kind of makes sense, but let's just try to get some data for it. So when you begin thinking about it, you look at Cryptococcus. By now, a large amount of data begins to emerge, and you can in strains. And we propose that, in fact, other groups have shown that the ancestor probably emerged in Africa, and this is what the world looked like about 100 million years ago. And the Gatti and the Neoformans, we propose, separate when the continents begin to drift. So that provides suggestive evidence that, in fact, Cryptococcus, since they are very similar, they both have capsules, they both make melanin, they both have urease, they both have all the goodies that you need for virulence, that there would have been virulence back in the time of the dinosaurs. David Engelthal and William Meyer, who know this stuff better than I do, actually took the same idea and superimposed on it all the cryptococcal types. And you can see then this idea acquires explanatory power, because now you can see why the isolates from Spain are kind of similar to some of the isolates from Eurasia, even though they are quite separate. So this is how an idea can begin to feed into experimental thought and explanatory power in other areas. And then in our lab, one of the things we did was, well, you guys know that we work on intracellular pathogens, that we torture macrophages with Cryptococcus. So we took very different strains that we know are separated by dozens of millions of years ago, and then asked the question, what is their intracellular pathogenic strategy? And the result is they all do the same thing to a macrophage. And that suggests that this story is ancient, that the capacity for virulence existed back in the Cretaceous, at least for this organism. I'm sure you can do it for Aspergillus. I'm sure you can do it for Candida species. This stuff was pathogenic back then. So then comes the second update, and the second update comes from my trip to the CFAR in Toronto. And they asked me to, as a lighthearted thing, to start the meeting with the dinosaur stuff and talk about this stuff. And then Chris Damon from the Gates Foundation comes to me at the coffee hour and says to me, what about food? Would mushrooms have provided food? He's thinking that all these mushrooms were growing on it, and that led to a lot of thought and a major update. And this has just been published this month, but the bottom line, this update deals with the differences between how mammals and reptiles would have coped. It turns out that reptiles would have been in big trouble, not only from fungi, but also from being cold. They need warmth to move. So looking for food would have been a major problem for them. They need warmth to digest their food. Their tract doesn't move if you cool them to the temperatures, the ambient temperatures of the time. And sex ratios are affected by temperature. So mammalian warmth would have a variety of other advantages. The mammals will have no trouble moving. The mammals will have no trouble digesting their food. The mammals would have then protected their offsprings in the placenta when they were warm. So the bottom line is that post-KT weather event conditions greatly favor mammals apart from problems with fungal diseases. So this third update now begins to look at the other factors that could have contributed. But again, the story becomes more solid the more you look into this, that our lifestyle, our temperature was selected for because in those horrible years after the cataclysm, whether your higher temperature protected you against infectious disease or allowed you to move, would have been a tremendous survival advantage. So the mammals proliferate, and then when the sky is clear, what you have is you have a lot of mammals and you have few reptiles. And these mammals now become the founder species for the great mammalian radiation that would happen in the subsequent few million years. So here it begins, this is an artist through it. You have the bolide impact with tsunamis, deforestation, you had the decline of the ectotherms, cold interference, reproduction, food acquisition, digestion, hypothermia. The mushrooms would have provided food for both reptiles and early mammals, but mammals would have been able to get them, reptiles would have had trouble. And then this leads to an expansion of mammalian numbers, and then you have the rise of the endotherms. And if you're interested in read, you can download it. It was published this month. I want to tell you how this one of thinking takes you to other places. The world is getting warmer, and the fungi can adapt. In fact, most fungal diseases that we know are near the equator, consistent with the fact that you need to have a lot of things to cause disease in a mammal. You need to have virulence factors, but you also need to be temperature adapted. And that's why not every organism, not every fungus that can grow to 45 degrees can cause disease, but you need to at least grow to 37 and survive in that temperature to be able to cause invasive disease in humans. And the other thing is that fungi can be trained quite rapidly to reproduce at higher temperatures. And this is a source of great concern to me, and because that means that you have a lot of organisms fully loaded for causing human disease that are being stopped from causing human disease by your temperature. But if you adapt them or if they adapt, they have now the capacity to breach the thermal barrier. And about 10 years ago, worried about this, we wrote this paper, which was just a warning that said that, there's a possibility this may happen. And then we have Candida auris. And the question is Candida auris the first of the fungi that break through? So again, it's a hypothesis. But one of the things I can tell you is that working with Vincent Robert, this is the CBS collection, and the first two lines are the ascomyces, which are largely temperature adapted already, but the basidiomyces are significantly more temperature susceptible. The basidiomyces appear to be adapting already. This is the average temperature of isolates deposited at the CBS collection for years, and you could see that it's even as the late 80s, the trend was up in terms of the new isolates being more thermally resistant than others. So humans can debate climate change. This data suggests that the fungi have begun to adapt decades ago. So for Candida auris, again, work with Vincent Robert, and he did analysis as to the close relatives, and the close relatives, which are other Candida species, are not thermally tolerant, or most of them are not. Occasional clinical isolates are. This organism was not known to medicine prior to 2007. It emerged simultaneously in 3 continents, South America, Africa, and Asia. And the isolates from the 3 locations are genetically very different. Since then, there have been other reports of other isolates emerging. And the question is, what's common to all these areas? Well, what's common is that the world is getting warmer. So people say, oh, Arturo, but it's only ½ degree. How could that be? Well, they're not. The way to think about this is don't think about average temperature. Think about the number of hot days. Every hot day is a selection event. So if you increase your number of hot days, every organism that makes it through that day needs to adapt in some way. And I think that is a selection mechanism. It's not necessarily the global world temperature. So we wrote this up, and this was published last year, got quite a bit of press. Again, a hypothesis. There is a lot of things here that can be tested. I think the most important thing is to find this organism in the environment. The prediction is that if you take it out of the environment, some of those are not going to be thermally adapted as the clinical strains. And it is remarkable how rapidly the clinical strains have adapted, even from the original isolates to the isolates that are coming out from patients today. So in summary, you are protected by twin pillars, and you have advanced immunity. By that is, you have B cells, T cells, and all the goodies that come with a very complex advanced vertebrate immune system. And we saw what happened. We saw that the rise in fungal diseases in the late 20th century have to do with immunosuppression. AIDS, cancer, drugs weakens this pillar. But now we have another problem. Climate change could weaken the second pillar, because as the world gets warmer, it reduces the temperature gradient between you and the environment. And also more organisms have to survive very hot days. And these organisms may be the source of new pathogens that are unknown to medicine. So I want to end with this idea. What you heard today is very different than when you hear in seminars. Generally, these are the ways what I call epistemic journeys. Epistemic is a great word that doesn't get used enough, but it comes from Greek, the knowledge. How do we know what we know? How do we acquire knowledge in science? There are a lot of ways to acquire knowledge. You can imagine that data leads to thought to theory, data leads to theory to thought, theory leads to thought to data. You can find examples of all this in the history of science, but these set of papers that I basically begin with thought. And they sometimes lead to data and then to theory, sometimes lead to theory and then to data. For example, you could argue that the Candida auris is going thought, theory, data. The data are not there yet. Whereas the case of the KT extinction began with thought, questions, then you look for data, and then you formulate a theory. But the final message I want to leave everybody with, and in particular the people of the younger age range, is that science should be fun. I went into science because it was fun. And people have been telling me all this time, focus, focus, focus. I will not focus. You know, I mean, I know how to do deep work, and I can do it in immunology, I can do it in microbiology, but I'm interested in a lot of things. And don't be afraid to think. Thought does not cost anything. You don't need a grant to think. It is okay to take risks, provided that you formulate them in sufficient caution and that you formulate them in established facts. And if wrong, you can always revise it. But I encourage everyone to think more and to try to make connections. And I see that I'm at 24 minutes. I'm going to take one more minute because I think that ending talks early is always good. But how do you, so I would say I encourage develop your curiosity. And this is like going to the gym. The more you learn, the more you want to learn. And in particular, try to learn outside other fields. People say, well, how to do it? Let me give you an example. Today, this month, 3 missions have been sent to Mars from 3 different countries. They have different scientific instruments. Look into what they carry. Look into how they make their measurements. Look as to what they're doing. And that just gives you, you begin to learn and you begin to put facts that way. I think that it could be a lot of fun. And despite, you know, we all get our grants turned down, we all get our papers turned down, but science should be fun. And with that, I end giving the group back 5 minutes. Thank you. So I'll stop sharing my screen. See, and here we go.
Thank you, Arturo. As always, a very exciting and thought-provoking talk. For those who are joining us more recently, we're going to be holding questions to the end. Arturo just gave us a few more minutes, so we'll have time for that. It is my pleasure to introduce our next speaker, Rebecca Drummond from the University of Birmingham.
Okay, hopefully you can all see the slide.
Yes, we can.
Great. Okay, so I recently set up a lab at the University of Birmingham in the UK, and I'm interested in trying to understand organ-specific antifungal immune responses. We do this to try and understand why some organs might be permissive to fungal infection while other organs might not be. And to do this, we study the behaviour of tissue-resident macrophages in the context of invasive fungal infection. So every organ in the body, as far as we can tell, is endowed with its own population of specialised macrophages. And these specialised cells are important for the homeostatic functioning of that organ. In some cases, they're also important for the development of the organ. But in adulthood, a lot of these macrophages also provide immune surveillance. So they're important for acting as a sort of frontline defence for any injury or infection within the organ. So my lab is particularly interested in macrophages of the central nervous system. So within the central nervous system, you have several populations of specialised macrophages, and these are typically associated with specific anatomical compartments. So today what I'm going to do is tell you about some of our published work which looks at the most numerous of these, called microglia, how they function in the context of a fungal brain infection. So microglia, as I say, they are these specialised macrophages found within the central nervous system, and in particular they're found within the brain parenchyma. They're the most numerous macrophages found within the CNS. They're also very long-lived. So this is quite unusual compared to other macrophage populations. The reason that they're long-lived is because they do not derive from monocytes. They're not, there's no monocyte turnover. They don't come from the bone marrow. Instead, developing microglia seed the developing brain during embryogenesis. So they come from the fetal yolk sac, and they then self-renew throughout life. And so that's why they're very long-lived. We know that they're important for brain development, and they do this partly by helping the formation of neuronal synapses. So that's one of the major roles during the development of the brain. But in adulthood, microglia seem to be particularly important for providing that sort of first-line defence against brain injury and also CNS infections. So this is a picture of one of them here. So they have this quite beautiful ramified morphology. So they have a lot of dendritic processes which they use to continually sample their microenvironment. And if they become activated either by a microbe or by some sort of damage molecule, they'll round up and form an amoeboid type morphology. So I'm going to tell you today about some of our work which looks at how these cells function in the context of a fungal brain infection. So fungal brain infections are thankfully pretty rare in humans, but these are the sort of three major sort of CNS infections we see that are caused by fungus. But most fungal, pathogenic fungal species can cause fungal brain infections. So the most common fungal brain infection is cryptococcal meningitis. This is typically caused by Cryptococcus neoformans as well as some other Cryptococcus species. And the major risk factors for that are prior infection with HIV and also medical associated immune-based suppression. Rarer is a CNS aspergillosis. This is caused by Aspergillus species of some kind. Again, always associated with immune-based suppression or neurosurgical complications. And this is also a particularly nasty disease with a very high mortality rate. Now, the disease that I've been working on in the last few years and what I'm going to talk about today is CNS candidiasis. So this is an invasive candida infection of the brain, typically caused by a candida species of some kind. And there are many risk factors for this. So like the other fungal infections, immune-based suppression, usually caused by some sort of medical intervention. CNS candidiasis is also a problem for neonates and low birth weight newborns, presumably due to the immature status of their immune system. But interestingly, it's also a major risk factor for CNS candidiasis is a primary immunodeficiency disorder known as CARD9 deficiency. And this is what I'm going to talk about today. So CARD9, for those of you who don't know, is a signaling adapter protein, and it's expressed mostly by myeloid cells, so neutrophils, macrophages, and dendritic cells. And it's coupled to many members of a innate pattern recognition receptor family known as the C-type lectin receptors. So that includes members such as Dectin-1 and Dectin-2. So these receptors bind to carbohydrates within the fungal cell wall. And upon that innate recognition event, they then initiate intracellular signaling pathways which utilize CARD9. And then this has the result of initiating a cellular immune response, whether that be to drive production of pro-inflammatory cytokines, but also to initiate fungal uptake and killing. Now, we know that CARD9 function is really important for activating a protective antifungal immune response in humans based on what happens to people who have deleterious mutations in the gene encoding CARD9. So that results in a disease known as CARD9 deficiency. And it's quite unique among the primary immunodeficiency disorders in that it uniquely predisposes you to having a fungal or one or more fungal infections. And what I mean by that, by being unique, is that there are many other primary immunodeficiency disorders, but we typically see a variety of different susceptibilities to infection in those disorders. So we'll see both bacterial, viral, and fungal infections, whereas card9 deficient patients, as best as we can tell so far, only have susceptibility to fungal infections. So we don't see any increase in bacterial or viral infections in these patients. This is an inherited disease, typically, usually autosomal recessive. So what that means is you have to inherit 2 faulty copies of the card9 gene in order to be diagnosed with this. And interestingly, with card9 deficiency, it's not a blanket susceptibility to any and all fungal infections. It's also quite restricted to the fungal species in particular, and also the organ that's involved. So card9 deficient patients typically present with very certain fungal infections. So for example, many of them will complain of mucosal candidiasis. There's a few cases now of card9 deficient patients developing extrapulmonary aspergillosis. These are aspergillus infections which do not involve the lung. But the 2 most common phenotypes that we see with card9 deficient patients, the first is fungal infections which affect the skin and subcutaneous tissue. So things like deep dermatophytosis and pheohyphomycosis. And the other common phenotype, and what I've been working on, what I'll talk about today, is the spontaneous development of fungal meningitis. And this is usually caused by a Candida species. And this really is spontaneous. So these patients don't have any other underlying risk factors. They're not having any surgery. They don't have any cancers or any other kind of immune-based suppression. So to give you a bit of background on what this looks like clinically, this is one of the patients who came to the NIH after being diagnosed with card9 deficiency. And this here is pointing out a mass of Candida cells in her brain, and we identified this as Candida albicans. But so she was around 10 years old when this scan was taken. And prior to that, her clinical history had been largely unremarkable. She had a few episodes of mucosal candidiasis, but otherwise was relatively healthy before developing a Candida infection of some of the bones in her spine. She had osteomyelitis, which then progressed into this infection that you see here. This is the same patient, another scan of her head. So if you're not used to seeing these types of scans, these white or paler areas here, these are the ventricles within the brain. And this is where cerebrospinal fluid would flow. Now in a healthy brain, these would be much, much smaller. The reason they're so enlarged in this patient is because that flow of spinal fluid is being blocked, and it's being blocked by Candida infection. And this obviously has a really big problem because this causes swelling and pushing of the brain tissue up against the skull. So in order to fix this, surgeons had to go in and put a shunt into one of these ventricles to help release the pressure. So this is a photograph from that surgery. So you're looking in the brain tissue here, and what I hope you can appreciate is there's this large mass of Candida albicans cells growing within the brain. So this is essentially thrush of the brain tissue. And this is despite treatment with antifungal drugs, so that clinically this is a very difficult infection to manage, and a lot of card9 deficient patients with this type of phenotype will succumb to their infection. Now this susceptibility to Candida infection, specifically within the CNS, is something in the context of card9 deficiency, is something that we can also model in mice. So if we take a wild type mouse and you infect it with Candida albicans, and we do this with an intravenous injection of Candida, those animals will get a brain infection, and this is typically what you see. So this is histology of the brain. So you typically see these small inflammatory lesions where the fungal cells are surrounded by immune cells. And based on the size and the shape of the nucleus of these cells, we can identify them as neutrophils. So you get this very early wave of neutrophils coming into the wild type infected brain, which seems to surround these Candida cells and very quickly contain and control them. So wild type animals will clear out a Candida infection from their brain very quickly, so within a couple of days. In contrast, if we were to infect a card9 knockout mouse, this is typically what you would see. So these are Candida albicans hyphae, so they form these filamentous forms, and it just grows uncontrolled. And what I hope you can also appreciate here, there's really no inflammatory immune response. So we don't see any of those inflammatory immune cells coming in here. And instead, the fungus just grows uncontrolled. And as a result of that, card9 knockout animals succumb to this infection very quickly, so they don't survive past a few days. Now this lack of neutrophils that we see in the card9 knockout mouse brain is something that we also saw in our card9 deficient patient. So in order to kind of get a sense of what types of immune cells were coming into the brain of our card9 deficient patient, we looked to see what immune cells were in her cerebral spinal fluid. So what we found there were lots of things like lymphocytes and eosinophils, but really no neutrophils at all. And that's in stark contrast to other patients who have wild-type card9 who develop this type of Candida brain infection. And that's usually a complication following neurosurgery. Those patients would have between 70 to 90% neutrophils in their cerebrospinal fluid. So the fact that she didn't have any, our card9 deficient patient didn't have any neutrophils in her cerebrospinal fluid, despite that high fungal load in her brain, was really quite striking to us. And it suggested to us also that this lack of neutrophil recruitment was probably responsible for the reason that you had this uncontrolled infection. So to test that theory, we went back to our mouse model and we depleted neutrophils and looked to see what the effect was in fungal brain infection. So we depleted neutrophils using an antibody called 1A8, and you can see this effectively removes all the neutrophils from the fungal infected brain, but it leaves something like inflammatory monocytes intact. And by doing that, you significantly increase the fungal brain infection. And so what that tells us is that that early wave of neutrophils that comes into the Candida infected brain is very protective, and it's necessary to help control that Candida infection. And it's very much dependent on card9 in both mice and humans. So what our next work wanted to do was to try and understand which card9 expressing cells were important for initiating this protective immune response in the brain. So we looked to see which cells were actually expressing card9 in the Candida infected brain. And what we found was that the highest expressors of card9 are actually microglia, so this long-lived resident macrophage population within the brain. So they express the highest amounts of card9 during Candida infection in the brain. So our hypothesis was well, maybe microglia are producing the signals that neutrophils then need to come into the Candida infected brain. So to test that theory, we then did a depletion model where we got rid of the microglia and then looked to see what happened during infection. So to deplete microglia, we used some transgenic animals where we could engineer expression of the diphtheria toxin receptor specifically on microglia. So this is based on a Creflox-based system where when you treat these transgenic animals with tamoxifen, this activates expression of recruitocombinase, which then drives expression of the diphtheria toxin receptor. And that's done under a cell type specific promoter to target it to microglia. And what then happens is when you give diphtheria toxin, this causes apoptosis of any DTR expressing cells, in this case microglia. So we made those animals and then we infected them and then looked to see what the effect was on neutrophil recruitment. So you can see that this model works very nicely. This is looking at the number of microglia in the brain. So the microglia deficient animals are in the open circles here, and these are their wild type litter mates. And you can see you can very effectively get rid of all the microglia in the brain using this transgenic mouse model. When we infected those mice and then we looked in the brain to look at the number of neutrophils, what we found is a significant reduction in the number of neutrophils in the microglia deficient mice relative to the microglia control mice. So what that tells us is that microglia are also critical in addition to card9 in providing whatever signals neutrophils need to come into the Candida infected brain. So in order for neutrophils to come into the site of infection or the site of inflammation, they follow molecular flags. So we call these chemoattractants, and they follow up a gradient of these flags to help them find their way to where they need to be. So these chemoattractants can either be chemokines, they can also be pro-inflammatory cytokines, complement proteins, and various other inflammatory mediators. And how important these are is very much context and organ specific. In some cases you'll get functional redundancy, in other cases you won't. So to try and figure out which of these was most important for bringing neutrophils to the Candida infected brain specifically, we took animals that were deficient in either of these mediators or their receptors, and then we asked the question, can those animals, if they're deficient in, say, CXCL2, for example, can they still bring neutrophils to the fungal infected brain, and can they control fungal brain infection? So in essence, we were looking for any mouse knockout that had the same phenotype as a card9 knockout. So we could try and map out which players were involved in this protective immune pathway. So the first mediator we found to be really important was a cytokine called IL-1 beta. So this is a pro-inflammatory cytokine that's processed via the inflammasome. So IL-1 beta knockout mice, which are shown in the purple dots here, when you infect them and you look at the number of neutrophils per brain, we found that that is significantly depleted compared to wild type control animals. And because those knockout animals are not bringing neutrophils into the fungal infected brain, they have a significant increase in fungal brain burden. The same is true when we infect animals deficient in the IL-1 beta receptor, which is called the IL-1 receptor, as well as the signaling adapter protein for the IL-1 receptor, MyD88. So both of these animals, again, we don't see any neutrophil recruitment coming into the brain, and as a result, you get this increase in fungal brain infection. The other major mediator we found to be important for this protective neutrophil recruitment was a chemokine called CXCL1, which is also known as KC. So CXCL1 deficient mice, again, have the same phenotype, produce neutrophils and increase fungal brain burden. And the same is true if you infect mice deficient in the CXCL1 receptor, which is CXCR2. And important to note here, CXCR2 binds several other chemokines, including CXCL2 and CXCL5. But what we're showing here is there's actually very little functional redundancy in this system. You really need CXCL1 in addition to IL-1 beta. So that allowed us to identify which inflammatory mediators were responsible for providing the signals needed by neutrophils to come to the fungal infected brain. So what we found so far is you need CXCL1, you need IL-1 beta, we know you need card9, and you need microglia. So our next question is, okay, so are microglia the producers of IL-1 beta and CXCL1, and do they do that in a card9 dependent fashion? And the answer is yes. So the experiment we used to test that question was we isolated microglia from the infected brains of either wild type or card9 knockout animals, and we re-stimulated them in vitro with either zymosin or LPS. And then we measured production of IL-1 beta and CXCL1 using intracellular flow cytometry. And what I hope you can appreciate here is that the microglia from the card9 deficient animals produce significantly less IL-1 beta and CXCL1 relative to the wild type controls. And so to try and really test the hypothesis that all you really need is card9 expression in microglia when it comes to protecting the brain, we made microglia specific card9 knockout animals. So to go over this model, the way that we do this is we took transgenic animals which have LOX-P sites outside of one of the exons of the card9 gene, and we call these card9 floxed mice. We then cross these to animals expressing the tamoxifen-inducible Cre recombinase under the control of the CX3CR1 promoter. So CX3CR1 is highly expressed by microglia, as well as some other smaller populations of monocyte-derived macrophages. So the way in which we make this model microglia specific is we treat our transgenic animals with tamoxifen. That activates expression of the Cre recombinase in any cell that expresses CX3CR1. The Cre recombinase acts on the two LOX-P sites to loop out exon 3. And that sense interrupts the expression of the card9 gene. So all those cells become card9 knockout. So when you treat with tamoxifen, you end up with your long-lived microglia in the brain being card9 knockout, as well as some short-lived monocyte-derived macrophages. Then what we do is we wait a minimum of 4 weeks before doing the infection and the analysis. And what happens in that time is monocytes start to replace these short-lived monocyte-derived macrophages. And these monocytes will never have seen tamoxifen, which means they are not switching on Cre, which means their card9 gene remains intact. Whereas in contrast, your microglia are long-lived, and so they will remain card9 knockout because they will still be expressing the Cre recombinase. So we made these animals and then we infected them, and then we looked to see what the effect was on neutrophil recruitment to the fungal infected brain. And so what we found was that the microglia-specific card9 knockout mouse, shown in red diamonds here, have a significant reduction in the number of neutrophils coming to the brain. So in essence, what we've shown here is by depleting or deleting the card9 gene in this one cell type in microglia, we can get the same phenotype as what we saw in a full card9 knockout. So by deleting card9 in this one cell type, you can really affect this protective immune pathway. And that has implications for control of fungal infection in an organ-specific fashion. So we see an increase in fungal brain burdens, but we don't see an increase in fungal burdens in an unrelated organ like the kidney, where we would not expect card9 expression to be interrupted based on how this model works. And we were also able to relate this to the behavior of the microglia. Because when we looked at microglia, again, by intracellular cytokine staining for IL-1 beta and CXCL1, we found that that was significantly reduced in the microglia-specific card9 knockout animals. And so that takes us to our model, where you have candida invasion of the CNS, and this is recognized by card9 expressing microglia. We did actually identify the fungal factors responsible for the card9 microglia are recognizing, but in the interest of time, I've not gone over that data, but you can read about it in our published work. So upon recognition of candida invasion of the CNS, these card9 expressing microglia produce IL-1 beta and CXCL1, and that provided for the neutrophils to come in, and then they're really the cells that then phagocytose and kill the candida, and thus help clear out the infection from the CNS. And so this is quite a complex multicellular player pathway, but we think it's what's protecting us all from getting this type of infection, and it fails in card9 deficient patients. And so we think this helps explain why card9 deficient patients have this very narrow spectrum of susceptibility because this protective immune pathway is failing in a tissue-resident macrophage, which is not found in other organs. And so other organs may be better protected because they're less dependent on card9, whereas microglia are very much dependent on card9. So with that, I'm just going to finish here with my acknowledgements. So I did a lot of this work when I was a postdoc with Mihalis Ioannakis at the NIH. So I'd like to thank him for hosting me in his lab, as well as all the members of his lab. We had many, many collaborators involved in that work, so I'd like to thank all of them. I'd also like to thank all the current members of my own lab at the University of Birmingham, where we are now focusing on how microglia function in the context of cryptococcal meningitis. And then all my colleagues at the University of Birmingham who've been very supportive in helping me get set up in my lab over the last year and a half. And of course, my funders as well. Thank you.
Thank you, Rebecca and Arturo. Those were both fantastic. So for those who just joined us recently, we're going to take questions from both of our panelists. We'll be alternating, starting with Arturo. And if you have questions in the audience, you can submit them through the Q&A. So Arturo, a couple of people asked about, they're interested in your thoughts about observations that human body temperatures appear to be falling and whether that might be a contributor to susceptibility to fungal infections.
Yes, I put this talk together this morning. I should have included that. But the answer is, of course, if you drop human temperature, you are going to even half a degree or so, one is exposing one to a larger number of organisms. It's very interesting that the idea is that in the past, we harbored a lot more infectious diseases, parasites, we were infected with TB, people were PPD positive, and a lot of things like that, which revved up the immune system and consequently revved up temperature. And it may very well be that, that as we get into a cleaner environment and less diseases, that we're going to be cooler, and that's going to cause us problems, potential problems with other fungi.
Okay. Rebecca, so there was a question about the mucosal candidiasis observed in the card9 deficient patients and whether that manifested as vaginal candidiasis, oral candidiasis, et cetera. If you could discuss organ specificity there.
My understanding is mostly oral candidiasis. I don't think there's been any increase in vaginal candidiasis in those patients, or at least not that's been specifically reported. Mostly oral candidiasis.
Okay.
Yeah.
So Arturo, the same selective pressures that would have been placed on birds, which are also
Yes.
warm-blooded and incubate their eggs. So have you looked at bird evolution at all?
Yes, they fit very nicely. The bird lineage is the only dinosaur lineage that made it through the KT boundary. The birds by then were already feathered and capable of flight. So presumably, so one of the questions that has always been around is why endothermy in the first place? And in the case of endothermy allows you, the hotter your engine, the more efficient you are. So if you are hot, you're able, for example, to produce the energy needed for flight a lot easier. But it doesn't make a lot of sense for terrestrial mammals. And the birds almost certainly became warm in order to maintain the height rate. And today they are susceptible primarily to aspergillus. Quite a few birds die of aspergillus, but aspergillus strains can easily survive on bird temperatures, which are between 40 and 42 degrees centigrade.
Okay. So Rebecca, there's a question, if you've looked upstream of the pathway to see if a receptor on the surface microglia that impacts the card9 pathway or this whole dynamic.
Yeah, so we took a lot of mice deficient in Dectin-1, Dectin-2, Dectin-3, Minkle, and some others, I forget. But basically what we found is there's a lot of functional redundancy upstream of card9. So if you knock out Dectin-1, presumably Dectin-2 takes over. And so we only saw a combined phenotype when we knocked out the Fc gamma receptor on a Dectin-1 knockout background. So we had to knock out all the receptors in order to see the same phenotype as the card9 knockout. So we don't think there's any one specific receptor on microglia that is activating that pathway.
Arturo, is there any evidence that we're isolating more different fungal strains now than we were 25 or 30 years ago? Meaning that due to warmer temperatures that these new species might be able to proliferate more?
So the CBS analysis shows that the isolates today, on average, are capable, have a higher temperature range than if the isolates that were isolated decades ago. And that is suggesting that at least what's coming out of the ground, what's coming out of insects, whatever you're getting out of the environment, it's already on average. I mean, it's consistent with. I don't think that there is a causal relationship here, but it certainly fits with the notion that these organisms in the environment are rapidly adapting already.
Rebecca, there's a question, was the effect of card9 deletion time specific? Meaning, did monocytes come into the brain after infection and replace the function of the microglia?
That's kind of a difficult question to answer because the card9 knockout mice die so quickly. I don't know if I can really answer that effectively. It's something we probably could have tested with our transgenic mice though, because we had a tamoxifen inducible Cree. So you could probably have come in later then. But that's not something that we actually tried, but it's a possibility because it's been known that monocytes can take on a microglia-like phenotype. But I don't know if that happens in a very short sort of acute infection model like this.
Sorry, I'm trying to catch up on the questions here. So there's a question, Arturo. There was evidence that the world was much hotter than now about 50 million years ago after the KT event. Is there any evidence that there were more fungal infections at that time?
So, you know, I have had paleontologists write to me and says, I won't believe this unless you can give me fossil evidence of fungal disease. I said, come on. You know, for you to get fossil evidence, it needs to affect bone because any part of the flesh is not preserved. So the answer is the evidence is certainly fossil evidence for more fungal diseases than is not available. The one thing we do know is that the world was 10 degrees warmer than it will get at the end of the century. There were forests in Antarctica. And the sea levels were much higher than obviously that they are today. So it was a warmer world, and clearly everything that lived at that time had to be much more temperature adapted than it is, higher temperature adapted than it is today.
A question to Rebecca, is the brain invasion specific to any particular species of Candida? Also, why is Candida mostly responsible for these infections and not other fungal genera?
So it's not one. So we've seen a lot of Candida albicans in carnine deficient patients, but also other Candida species. So I believe Dubliensis has been one that's popped up quite a few times as well, and a couple of others. So it's not specific to one particular species. Why it's specifically Candida is probably to do with the fact that the fungal factors are being recognised by card9 expressing microglia. So we found it was the toxin Candida lysine, which is being expressed by the Candida cells, and that's being specifically recognised by microglia, although exactly how that recognition is happening, we don't know if that's receptor-based or damage-based, for example. So that could be a reason why it's Candida specifically. But again, we don't know how card9 patients actually get Candida in the brain in the first place. That's a completely unanswered question because presumably it doesn't just land there randomly. So maybe something to do with exposure, for example. But again, these are all unanswered sort of questions within the clinical field.
Okay. Arturo, so question, you've developed the theory that fungi contributed to the mammalian era, but what do you think contributed to the appearance of fungi? And there are theories that at some point fungi were the dominant life form. Considering this, do you think temperature would have been the main factor in changing this scenario, or would you argue they're still the dominant life form? So you can select what you will from that question.
You know, this one is outside my league. I will accept that they are the dominant life form today. I don't have any problem with that. But they are mostly microscopic, obviously. But I think that is fascinating, and I think this kind of thinking, whoever posed the question, I encourage you to spend some time with it, because once you begin thinking this way and you begin thinking about deep time and you try to begin connections, things are a lot more fun, are fun. And you don't have to apply for funding for this.
Always a
You
good.
don't, you know, it's wonderful. You know, your grants won't get trashed. You know? But if you find one molecule and you try to apply for funding for it, you'll get trashed.
Rebecca, there is a question about whether individuals who are heterozygote for CARD9 have phenotypes.
Not that we're aware of, no. So a lot of these CAR9 deficient patients we see, they have siblings who have who are heterozygous, and that's typically because it's consanguineous marriages usually that results, because it's autosomal recessive infection. But I haven't seen any reports of that, and at least the CAR9 heterozygous mice didn't really have much of a phenotype either.
Okay. So for Arturo, how have existing reptiles overcome fungal selection pressure?
They have problems with fungi, but what they do is they sun themselves. If provided that there is sun and there is a hot rock, the reptiles and the insects will kill off the fungus with heat. In fact, that study that I showed you about fungal adaptation was the result from a company in which they were trying to the problem is they have a fungus for killing insects, and the insects know to find a hot rock and to kill off the fungus with heat. So what they do is they were trying to adapt the fungus to a higher temperature so that the organisms can't. So bottom line, if you're not an endotherm, you get yourself an adaptive fever. You find something hot, you move a lot, you beat your wings, you raise your temperature one way or the other.
Oh, that's really cool. To Rebecca, there are several questions about implications for therapy. For example, giving exogenous CXCL1 or other strategies like that.
Yeah, I mean, I'm not a clinician, so I wouldn't want to sort of recommend anything, but that's definitely a strategy that was being discussed. One strategy we had thought of was using GM-CSF. So GM-CSF has been used successfully for some CARD9 deficient patients. And so we'd had the hypothesis that maybe that was because it was stimulating neutrophil function or recruitment. And so we actually treated that CARD9 deficient patient whose scans I showed you with GM-CSF, but it didn't make her better. It didn't reduce the fungal load at all. And so we published a paper on that in the Journal of Allergy and Clinical Immunology. And what we think is happening is it's dependent on the type of CARD9 mutation you have that's resulting in your disease, whether or not you respond to GM-CSF, but it doesn't appear to be affecting any neutrophil recruitment. So even GM-CSF treatment didn't affect neutrophil numbers at all. So yeah, again, yeah, it's a possibility, but it's not something that we've looked at specifically.
Arturo, there's a question. Are mammals evolving fungal-specific hydrolases or other soluble antifungals?
I don't know. I don't know the answer to that. I think that we as mammals are not under great degree of mammalian of fungal selection, because your average mammal, except for dermatophytes, or skin infections, really doesn't have a major problem with fungi, provided that your adaptive immunity stays in place. So I don't know the answer to that. I think that's an interesting question to compare them across evolutionary scales.
Question for Rebecca: is neuronal peptide signaling altered in the CARD9 deficient, I guess, mice or people? Any evidence that neuronal signaling assists with immune cell homing or activation?
I've no idea, not looked at all. It's an interesting question for sure. I think there's probably going to be a lot more cellular interactions going on in there than we're currently aware of.
All right, so to Arturo, is there data showing fungal isolates thermotolerance over time for specific geographic regions or locations?
You know, that hasn't been done. I think it's possible that I don't know if they have enough samples to look at that, because once you begin limiting, for example, to isolates from Italy or so, your power will drop remarkably. But I would say the people in this audience that is a testable thing if you, for example, begin saving isolates and are willing to wait a while.
Question to Rebecca. Why, I think you may have sort of addressed this a bit, but why these patients with CARD9 deficiency develop spontaneous Candida infection in the CNS? And does the Candida tropism change in this context?
I think that's an interesting question. My suspicion is it's probably that the Candida maybe escapes from the gut possibly and goes to all different places, but the other organs are able to clear infection because they're not as dependent on cardinine, whereas in the brain it doesn't. And so that's why it presents there. And if they're having a very mild infection in other organs, you might not see it because you might not get any symptoms. So the patients might not report it, but you will report some of these CNS symptoms because they can be quite severe, as I showed you with the scans of that young girl. So I doubt that the tropism is significantly changing with the Candida. I think it's probably got more to do with the sort of background susceptibility of the individual organs.
To Arturo, how does the rise in temperature alone explain the parallel evolution of Candida auris in 4 different continents?
So what I would say to you is that the organism was sitting out there, wherever you want to place it, in swamps, in birds, or whatever. And it was in a not temperature adapted. I mean, if it was on the birds, it would not be internally because the internal temperature of the birds is much higher than ours. So, but it was loaded. It was capable of defeating a mammalian immune system if put in the right conditions. And then what happens is that in 4 different places, you basically put this organism through a temperature selection by having more hot days that it needs to survive. And then somehow some of these organisms get into the healthcare system. Whether they came in hands, they came in food, I don't know. But once they got in, they were loaded already to cause disease. And now they had the temperature range. And then you begin to see the emergence of disease initially in very susceptible patients, people that were immunosuppressed after having surgery or so. But now that it's pretty much adapted to the hospital environment, we will have to see whether we get further trouble from it.
So to Rebecca, do the microglia need to engulf Candida albicans, yeast, or hyphae to produce and secrete IL-1, or is the receptor ligation enough?
So for the IL-1, we needed activation of the inflammasome, so we showed that. So presumably, because it's candilysin, which is a toxin, so it causes damage to the microglia, and I suspect that was probably what's activating it, so you're getting sort of inflammasome activation due to that damage. For production of CXCL1, what we found is that that candilysin recognition by microglia wasn't enough. What you actually needed was interactions from another cell called an astrocyte. So astrocytes are another type of glial cell. They're not myeloid cells, but they're found throughout the brain. So there needed to be some sort of cellular crosstalk there between those 2 cells for the CXCL1 to be made. So I don't know if you need to have full phagocytosis of the Candida itself, or whether, because the candilysin is a soluble factor. But that's not something that we tested specifically. But my feeling is that it would probably be a sort of damage-related signal that's causing that through the toxin.
After Arturo, there's evidence that for increasing aggressive fungal pathogens in reptiles, particularly in the genus I can't even read these. Nanozypopsis and Ophidiomyces, even though they're seemingly been around for a while. Could we be seeing a sudden uptick of physiological changes in such fungal pathogens? And has anyone
what may be happening to the reptiles is that they may be losing their one column, which is adaptive immunity. With the steroids that are thrown out, with the chemicals that are thrown out, there has been a lot of evidence that a lot of these organisms are affected. So what you may be seeing is that they only have one column, which is adaptive immunity. And as a result of pollution and the discarded chemicals that humans have put out, that they are becoming immunosuppressed.
Rebecca, does cardinine expression reduce in the aging brain, and is there any evidence for sex-linked regulation of cardinine?
I'm not aware of any studies, and I haven't personally looked at cardinine and how that affects in the aging brain, although there are a lot of published data sets now looking at microglia throughout the mouse's lifespan anyway, so it could be quite interesting to go and look at that and see if cardinine transcripts fall, for example. And I think there are some sex-based differences for cardinine function, but in other contexts. I don't know if that seems true for fungal infection. I think in some cancers, for example, that's been shown.
question for Arturo about fungi and plants and the radiation of the plant species post-extinction.
You know, there I don't know very much, so I don't even want to comment. But that's something that be good to look at.
So for Rebecca, how about patients with autoantibodies to GM-CSF?
So I know that people with autoantibodies against GM-CSF are more likely to get cryptococcosis, so cryptococcal infections of the brain. I don't know if anyone's linked that with Candida infections of the brain, though. And as far as I know, there's not been any description of cryptococcal or increasing susceptibility to cryptococcal meningitis and card9 deficiency. So I don't know if there's much of a link there, but then I don't know of any studies that have really specifically tried to go and look at that on a mechanistic level. Yeah, so that's about all I can say on that, I'm afraid.
So Arturo, there's a question about the temperature of dolphins and other marine mammals.
They are 37 or around. They are around our temperature. The marine animals are quite advanced. The mammals, the dolphins went back into the water. As you know, animals basically colonize land, early life forms, and then you had the great land expansion. And then these mammals basically return to the water long after the KT boundary. The one thought I had listening to Rebecca and to some of the immunological questions, and I think, and I give that to the immunologists, is all the work that we do is at 37 degrees. But most of the important interactions between immune cells and fungi occur at much cooler temperatures. The temperature in your hands, the temperature, for example, if you get a cut or something like that in your nose, is much cooler. And there has been almost no exploration of temperature and immune function, especially cells. You know, how does the macro, you know, you could do it because, and this is, I think is an area that is very interesting and potentially ripe for new insights.
Rebecca, I'll just read this question verbatim. So there are different chemokines and cytokines that can recruit an innate response to candidemia, for example, C3A, IL-1 beta, CXCL1, depending on which organ's affected. What would be the reason for this? Is there an advantage to the organ-dependent recruitment redundancy?
That's an interesting question. I'm not actually, I don't know if I could answer without, you know, checking. But I mean, my feeling was always that the reason that it was so specific to IL-1 beta and CXCL1 R models, because those were the ones that were card9 dependent, and you needed card9. So I mean, you're still getting production of CXCL2, for example. It's just because that's not so dependent on card9. And so that's why it was particularly those chemoattractants in that context. And the same might be true for you know, other signaling mediators in other contexts. Because I know people had looked at card9 expression in arthritis, and it also appeared to be sort of limited towards things like CXCL1 and things like that in that condition as well. So probably more to do with dependence on the signaling mediators that actually drive the expression of these mediators, rather than the organ dependency, if that makes sense.
So for Arturo, considering the difference in genomes between the Orris clades with a blanket increase in antifungal resistance, could temperature adaptation be playing a minor role only? With antifungal use in origin countries driving the pathogenicity, since Orris isolates can grow up to 42 degrees as well as above 37?
So yeah, I mean, some people have proposed that the rise of Orris was the result of selection from azoles. I mean, I have trouble imagining how just becoming azole resistant, which is the major selection in the environment, will give an organism the capacity for virulence. I think the capacity for virulence has to be very complex and multifactorial, and it has to be there probably for capacity for other organisms. So I won't rule out, for example, that azoles could have, if you think about the work that Leah Cohen has done, dial up for capacity to resist stress, and that in fact enhances virulence by an indirect mechanism. But I tend to think that the temperature is a more important driver than azoles, but then again, that's just an opinion with no data.
Rebecca, is card9 ubiquitous across animals?
If I remember correctly, yes, it's found quite sort of distant, sort of going evolutionary back. It's more the receptors that are only found in sort of higher mammals, which kind of makes sense because it's sort of the sort of hub of the signaling. And then, you know, higher mammals then develop these other sort of signaling receptors to kind of get the sort of more nuanced signaling pathways. But card9 is found quite far back, if I remember correctly.
Arturo, can thermotolerant fungi hybridize or cross with non-thermotolerant fungi to pass on their thermal tolerance?
That hasn't been done that I know, but it's very easy to generate a temperature deficient mutant, right? All you really have to do is to find a protein that is critical and put in a temperature label mutation. Going the other way is a lot harder. That is that you need to train, and you need to put in a lot of systems in place to resist higher temperature, because higher temperature is inevitably associated with more cellular damage. So it is a great experiment if somebody has two strains that are, what is the genetics of thermal tolerance? I mean, if you have two strains that can mate and you have one that is not thermal tolerant and the other one can grow higher temperatures, I think that's a very interesting experiment to do. And I hope that that's the kind of stuff that comes out of a seminar like this. I'm not aware of that having been done.
Rebecca, the brain fungal infections limited to clearance by the innate immune system, or do T cells and other aspects of adaptive immunity come into play?
So in the context of candida brain infections, my suspicion is it's like mostly going to be neutrophils that are doing the heavy lifting, where monocytes, I'm not sure, and they might also be involved, but we haven't really tested that. But neutrophils seem to be absolutely critical, I think. And other fungal brain infections, so I think the question's still open there. So cryptococcal meningitis, for example, is often associated with lack of T cell numbers or function. So they might be more important for that infection, whereas neutrophils seem to be more critical for candida. So it's probably going to be fungal species dependent.
Arturo, with the rapidly adapting isolates that are appearing today, are there more Ascomycetes or Basidios?
Basidios. The trend seems to be the Basidios. And that makes sense because they are more thermally susceptible than the Ascos. So the Ascos, the majority of them, are already in good shape. They can take it. They can take the changes, but the Basidios have to adapt. So most of the changes are with Basidiomyces.
Okay, I was just told that we are scheduled to finish in actually 3 minutes ago, but we're going to let each of you have one question each, very short. So quickly, Rebecca, do you think CX3CR1 positive monocytes also play a role?
Again, we haven't really checked specifically. I think in the context of candida, as I say, I think it's neutrophils that are probably doing most of the heavy lifting, but in other fungal infections, it's quite likely that they are also going to be playing a role. Particularly in crypto, I think that's been published quite recently, that those types of patrolling monocytes are probably contributing towards pathogenesis quite significantly.
All right, and so final question for Arturo. Arturo, in Cincinnati, we have lizards that are running around everywhere in summer, but survive our cold winters no problem. When the temperature begins to drop, what are your thoughts on how they survive exposure to fungal saprophytes?
Well, some of them manage to go on the ground, right? And with the temperature, but generally, as it gets colder, they need less food, and if they need less food, they can survive for longer simply because the metabolism slows down. I don't know in the city itself, but we in our environments we have created all kinds of different spaces where animals can survive simply because we keep our buildings warm. That's something to think about. I wanna, my last words is to say that congratulate Rebecca on a great talk. That's terrific. And to say that the hardest job here was Sarah, because she's got to watch the chat stream, she's got to moderate, she's got to change and I want to thank you for a terrific job.
Well, thank you very much. Thanks to both of you for fascinating talks. There was a lot of kind of cross-fertilization, I think, coming out of the questions. Many people are saying thank you on the Q&A. And thanks, Arturo, for recognizing the juggling act here. I'm going to go back and watch this on YouTube so that I can actually focus on your science. And so for the audience listening, this will be available on YouTube. I don't know exactly when it's going to go live, but it should be fairly shortly. And we will be having these once a month. Please check the website to sign yourself up. And tell your colleagues, tell your friends, share on Twitter, and everybody should stay safe out there. Wishing you the best. See you in a month.
Bye-bye.
Thank you.